Um, Hello, everyone. So we will be starting the webinar now. So good morning, everyone. Today we are here for the 20th edition of the CPHMS webinar series, which is on developing human relevant research in India. And today we have Dr. Gitajali Yadav from the National Institute of Plant Genome Research. And just before we begin the webinar, I would like to take you through what work we do here at CPHMS. So let me just go through that for a minute and we can get started with the webinar. Okay, so CPHMS, as in Center for Predictive Human Model Systems, is an initiative supported by Atal Incubation Centre, CCMB. And we are a science and policy think tank, and we were established in 2019. Now, our focus is basically to enable new technologies and model systems which are based on human biology. So systems like organoids and organs on chip, in silico models built using human relevant data, 3D engineered tissues, and even advanced technological techniques, advanced imaging techniques are systems which we promote. Basically, these systems can generate human relevant data, which can capture and predict the subtleties of human biology. And we want to promote these systems rather than relying primarily on animal models. So thus our main aim is to prioritize investment and research in India on human-based non-animal methodologies. So far in our work, we started with increasing awareness, in which we have published many popular articles, reviews, and we conduct these monthly webinars. That also comes in our education and training section, where we have conducted workshops before. Monthly webinars are a part of this series. And we also have an India Embo lecture series coming up. So we have also made policy initiatives where we have so far published 3 white papers, which were on developing adverse outcome pathway frameworks, developing systems biology research, and using microphysiological systems as a model system in the Indian scenario. Now, we have also published a perspective paper, and we have conducted multi-stakeholder roundtable meetings, which were conducted to provide recommendations to the government bodies. We also have conducted global collaborations, and we have also so far provided funding to Indian scientists who are working on the adverse outcome pathways in the field of cancer. So as I mentioned, the India Embo lecture course, we have this India Embo lecture course coming up, which is on microphysiological systems. We have researchers from all over the world who are working on systems like organoids, organs on chip coming up. And this will be an in Hyderabad in-person event conducted in October. We have currently closed the registrations, but we're quite excited for this lecture course. And in creating awareness, we also have our CPHMS quarterly newsletter. The link will be pasted in the chat box. So in this newspaper, in this newsletter, we go through the recent advances in the fields of human relevant research. And we are also trying to create an interactive database of researchers all over India who are working on human relevant models. And we are gathering this data, and this data will be an open access database, which will be published on our website. And we have a link for all the researchers to contribute to this database so that we can have them in our database. And thank you so much. You can read more about CPHMS on our website. And with this, I would like to hand over to Dr. Gitajali Yadav, and we would like to start with the webinar.
Thank you, Kadambari. I'm going to try and share my screen, and
Dr. Yadav, you're not audible.
Okay. Can you hear me now?
Yes.
And can you see my screen?
Yes, looks good.
Okay, thank you. And a big hello, CCMB. Kya haal hai aapka? So I am pretty glad to be here with you guys today to talk to you about a topic that I particularly love because I've been teaching network fundamentals for years and years, but more importantly, I've been doing research in network biology for a very long time. I think I began somewhere at the turn of the millennium. Oh, no, somewhere in 2007, okay? So about myself, I work at the National Institute of Plant Genome Research. I've been here 17 years now, but I'm also a professor at the ISO Bhopal for the last 2 years, and I'm affiliated with the University of Cambridge on a DVD deputation. So there's multiple avatars that one has to juggle between these days, and I think that's actually a good thing for interdisciplinarity and for not getting locked into these little silos that we find ourselves in all the way from being a student. So this is a general scope of the kind of work that I do in these three places. My bread and butter, of course, is genomics, transcriptomics, and this large amount of, constant kinds of seq data. RNA-seq, DNA-seq, MNA-seq, ADAC-seq, ChIP-seq, God bless you, seq coming in. And somebody has to be looking at that and trying to make sense of it by reducing data dimensionality. And that's what we do in the lab on a daily basis. We also apply our insights to structural models in order to try and see how ligand recognition happens in active sites. So we need to understand active site architecture, as you can see in one of the pictures here. But lately, in the last 5 years, I have become personally interested in biodiversity informatics, and so change is the only constant, and I hope to steer my lab into research on climate change as we go forward. And for that, of course, I'm already looking at nectar chemistry of certain species. We're looking at trees outside of forests, particularly in India, as part of one of the UN SDG programs on grasslands and urban trees. And living in a city like Delhi or a cosmopolitan town, you do wonder what happens to these poor fellows, the trees growing around us with these mighty sodium lamps shining down on them at night, and nobody's looking at. A lot of plant scientists, even you guys must be thinking about, stresses of 10,000 kinds when you think about the organism you're working with. Nobody looks at ALAN stress, that's artificial light at night. How often is your circadian cycle being disrupted because you've been chunked out on your head with all sorts of lights. And of course, we're moving into invasion biology, trying to understand what else we might do for climate. I read bad news, right? You start in the morning and you find out today that 180 acres of rainforest are going to be wiped out from Great Nicobar Islands in India, where we couldn't even go without a permit in the olden days, and when we were students, you needed a permit, and now we're going to wipe it all out to set up a mighty transshipment port. Of course, that's a good transshipment port. Of course, it's going to bring in trillions of dollars for India. But that heartbreak that you get, that not enough of us are thinking about what we are doing to what we have. So that's another story altogether. And of course, अब तो China ने श्रीलंका में बिठा दिया है अपना port, तो हमें कुछ ना कुछ तो करना ही है. So of course, all that is very important. Anyway, coming back to my work, I work on algorithm development because I'm a computational biologist, and most of the work we do in genomics, transcriptomics, biodiversity informatics has always got an angle to being able to develop tools and resources that the scientific community can use. And so we've been doing a lot of network biology, which I hope to explain some of it to you today. But network perturbations are our interest. Building web servers and resources for people from our work is our interest. And in the last 2 years of the pandemic, all of this masala that I've been doing has got another angle to it altogether, where I think we moved into, and more importantly, the priorities shifted during the pandemic, and I moved into trying and capacity building. Now, before the pandemic, I was doing some outreach, but it wasn't enough. But during the pandemic, I did realize that, unless we stand up, so there's something called the scientific social responsibility, and your students, everybody's been locked in. So I started doing these things called the what did I call them? Protocols from home. And on weekends, I would teach people data skills, and students from all over the world, whoever is interested would be willing to come in and work, listen to me blabber around about what we do on a daily basis and learn some skills. Even now, I keep typing out to my students, keep apne friends ko bulao aur sikhao, ya fir mere se jab main lecture de rahi hu to sunne ke liye bula lo. That it's very important that you as biology researchers have got to be aware of data science and to know what to do. So we've been doing trainings and internships, and this talk today is also part of that effort of mine, that every time I get an invite, in the olden days, because it used to be physically in person, I used to say no so many times that I don't have the time to travel, I can't visit you, I can't give a talk at your university. But the pandemic changed that, and the digital world today allows us to give these talks while we're sitting right here ensconced in my four walls. And so therefore, I'm again very happy to be able to do some capacity building without having to move out. And I'm working not just within India, talking to you guys, but also working for the WHO, where we are trying to help public health officers in different WHO, CRs nations, Southeast Asian nations, to understand what are the, walks and voice, variants of concern and variants of interest, and how to identify them using genomics for the SARS-CoV-2, of course. And there's a lot of work going on in the Asia Pacific as well. Okay, so today's talk is part of that. But during these 2 years, another thing that's happened very interestingly is that, in India, and that's something we all know, in India, research is not postdoctoral research. Abroad, outside of India, all of the lab, all of the research, all of the cutting edge research gets done by postdoctoral scientists. Okay, in India, that's not it. In India, our strength is PhD students. But what I discovered in the last 2 years was that we have a group of, interns. We could get interns from different places. And it started with the pandemic, but I've been able to grow this network of interns. It started with my collaborator at Cambridge, Peter, and we've been able to grow this huge network of students who come and join us for a few weeks, for a few months sometimes, and they just ran and got the, United Nations SDG Accelerator Award this year at the University of Geneva. And when they got the first prize, my eyes were like, oh my God, maybe our research can also be run by, these young students. So young scientists, people like you, are the ones who are going to change, the way we do research going forward. And I have huge faith in being able to do good work in a short amount of time, particularly if you're doing, data science and computational biology. So welcome to that again. And this is what our students have developed, something that we want to showcase today, how we extract knowledge using AI. And we've automated review of literature that you have to do. Each one of you in your research, you have to do review of literature, but you can automate it by using the tools called Semantic Climate that we've built. And we're going to be showcasing it today. You're all more than welcome in the afternoon to come and see how we do it, a 6-step auto chunk to get this done. So all that aside, thanks to my interns. Coming back to what I'm going to do today, the first 5 minutes, which I have already taken, are going to be on the quick overview and introductions. I have introduced myself. The CCMB people have introduced themselves. Now I'm going to talk to you about network concepts and background. We will take a short case study, if time permits, on PPI networks, and I will talk to you about how to represent networks, basic topologies, layouts, and maybe, if time permits, the concept of network partitioning and clustering. Finally, with some tips and tricks on looking at your data as a network, okay? If at all you have any Q&A or questions for me, don't wait till the end. There may not be time. You can type them out into the chat box now, and I'll be happy to take those when time permits. So concepts and background, right? There are diverse kinds of data sets in biology, and even more so now in this era, in the 2020s going forwards, when everything in the universe is digital, okay? So your data may be spatial. Close your eyes. Take a moment, think about your data, not mine, not what I'm telling you. What kind of data does yours look like? Is it spatial? Is it tabular? Is it textual? Is it raw? Is it unstructured? Whatever it might be, you can think about visualizing it as a network and be able to gain more insights from it than you are doing now if you're not using networks, okay? You might be working with genomic data, proteomic data, microarrays. The purane ho gaya. I don't know why this slide has microarrays. Nobody lives in that's kindergarten. Even if somebody mentions microarrays, run from them, because now we're on to high throughput sequencing in next gen and double next gen. So you have got all these different seq data sets, and the bioontologies are there, enzyme structures, reaction chemistries are there. All of you would be having all of this in whatever organism of interest that you're working on. But are you able to look at them all of these data sets together? Or are you first looking at the Excel sheet, which has the names of genes, then the transcript IDs, and then the condition-based transcript abundance, and so on? And if you're doing that, you're still sitting in the Victorian era, and you need to wake up and learn some data science, and particularly network biology. Okay, so for example, you just heard the series of talks is about those of you who are interested in human research, a multicellular higher organism, an animal not to speak of. I'm working with a unicellular species sitting in front of you, this little beast called clammy. I'm working with clammy. And do you know the kind of data I have for the single-celled single-cell eukaryote? I have typed out at least 15 different data sets, and these are all high throughput data sets. So if for the single cell, I have 15 different high throughput data sets, what might you have for the human beast? And so therefore, once again, it's very, very, very important for you to be aware of what you don't know. Ignorance is not bliss, right? So there are many different kinds of networks in biology, and complex biological systems, of course, can be represented and analyzed as networks, and this whole talk is for that. You might be thinking about ecological food webs or species interaction networks that we are always looking at. In my work, we're looking at plant pollinator networks, seed dispersal networks, root frugivory. But you would be looking at neurological networks, metabolic networks, signaling networks, genetic interaction, GRNs. We're looking at GRNs far more, because these days a lot of focus with the high amount of, I don't want to call it big data because it's not truly big data, but it is far, far, far bigger data than any that we've seen in the past, and that is the transcriptome data sets. And so from the transcriptome data sets, how to extract regulatory inferences or to develop regulatory landscapes is a major theme of all genomic omics research that's happening presently today. And you wouldn't be able to extract that information if you did not understand network science. Okay, PPIs, protein-protein interaction networks, are the simplest kinds of networks, and those are things I'm assuming you guys are postgrads, or are you undergrads, or are you postdocs? Just type out in the chat box, and I would like to look at it sometime later to get an idea about what kind of audience I'm talking to today. It's good to talk to strangers, but good to know who is there listening to you. There are other kinds of networks in biology, co-expression networks, co-annotation networks, drug ligand associations that probably you all are more interested in. Does anyone know any other kinds of networks? And if you do, why don't you type it out in the chat box, and I'll take a look at the chat box. I think I'm going to open the chat box at the other side. Okay, thank you, Joshi and Lavneesh. That's nice of you to answer me. Then, of course, the network analysis, the whole idea is not new. Just because I started working on it at the turn of the millennium or 7 years later does not change the fact that it was 300 years ago when the set of abstract concepts and methods were used for the first time by there we have, in a city called Kaliningrad. And the rule was, at that time, the question was that you have to use the 7 bridges in the city, and you had to cross all the 7 bridges without touching any bridge twice, and you couldn't leave any bridge also. So Leonhard Euler in 1735 solved this puzzle for the city and for crossing the city. Can you find the solution? The city's right here. The easy map of the bridges of the city is also on the screen, as built by, as interpolated by Leonhard Euler. And this solution was the one that set the basis of graph theory going, and the whole idea of topology, of looking at a picture or a map and converting it to a topological representation, became, was in place in 1735. So we are all coming very, very, very late into the business, okay? But it's good to be here, better late than never, right? So it was Euler first, and then for 200 years, nobody looked at networks. Then, of course, people in social sciences, psychologists and sociometry folks realized, anthropologists in the 1900s felt that, man, I can understand far more about the data that I have. And they mostly used to have survey kind of data answers, I wouldn't say subjective data. And from that subjective data also, they could glean out insights and knowledge by using networks. And yet you can still see life scientists have not yet jumped out a bulb. I mean, we are all biologists, but we are not the brightest bulbs on the Christmas tree, okay? So the sociologists rose in the 1970s. And then, of course, the IBM ka pehla PC was invented, of course, and network programs came into being in computer science first in the 80s. Tab bhi hum so rahe the. Then, of course, 1990s happened. And finally, at the turn of the millennium, physicists descended upon the network science. And if you look at the archives today, you will find 90% papers have networks in one form or the other in the archives. And that too is not life scientists mostly. We are bioarchive people, right? So we came in a little bit later. Okay, I still think that I came in sooner than a lot of other biologists around me. And I'm going to drag many of you with me if I can, okay? So what is a network? A network is a couple of nodes and a couple of edges. The nodes could be a set of actors, you and me, people, genes, proteins, genomes, collections, species, countries, just about anything. And these actors are all connected in a network by these edges. And we call them edges, but the edges are intentionally relationships. And the relationships are the kind of ties that connect these actors or the pairs of actors, okay? And so the kind of network you're looking at will usually be defined by the kind of relationship of tie that you have between these nodes. So you could have positive ties, you could have negative ties. I'm speaking here, but you don't get a single penny and it's all passing over your head. So we're having a negative social interaction right now, which I'm not aware of because I feel it's a positive one. Or it could be, acquaintance network. If the relationship is that you know me and I know you, or we are friends, then it's a friendship network, like on FB, right? If the two of us have published a paper together, then we might be connected nodes on a co-authorship network, and so on. All right. So why should you be thinking about networks? Because they are everywhere. Molecular networks, communication networks, social networks, nervous system, and they're very, very powerful tools. They're far more efficient than tables. As I keep telling my students, chuck Excel. You can't be touching Excel if you're a PhD student, and if you're even opening or clicking it, you need to go and jump from some kind of something. Anyway, coming back, it's great for data integration. You can have intuitive visualization, particularly and more so in biology. Very nice, Sonali. Autocrine, paracrine, endocrine networks, fantastic. Cytokine networks. And yes, these are indeed, as I had mentioned, Abhijit, I wrote about the drug interaction networks. Tell me something that I didn't write already. I need to add to that slide every time I give the talk to some new people, okay? So it's not just, so why should you care about networks? You need to care about networks because it's not just the elements of a system that matter, but also how these elements are put together, okay? And you can integrate, as I said, a large number of diverse data sets meaningfully. Now, on one Excel sheet, spare me. On one Excel sheet, you have the proteomic data. On another, you have the metabolomic data. On a third, you have the annotations. On a fourth, you have the gene names. On a fifth, you have godforsaken something else. But to be able to integrate them meaningfully, you don't do Monday ko ye wala Excel, Tuesday ko wo wala Excel sheet and pour your whole day. And after that, you someday die of old age. You can do that all together in 1 minute by being able to, in a few minutes at least, by being able to integrate all of these sheets, Excel sheets with each other and chucking them away, save as CSV, and bring into a set of tools that can allow you to visualize your data as a network. I will talk to you about those tools also. And you can quantify relationships between variables and individuals. You can gain insights at the smaller picture as well as the bigger picture. So you get to stay within the box and get out of the box if you wanted to. And you can find patterns and create new hypotheses. All of life for a scientist is about reformulating the problem that you're looking at, right? So coming up with newer questions or newer lines of research that you might go into. Networks can help you find those rationally. This is my student C2. She has just completed her PhD, and she was working on time series data. A lot of you would have time series data potentially in your areas, in your work. And she's been able to extract gene regulatory networks from the time series data of the circadian cycle of this single-celled eukaryote that we are working on. And she divided that into the cell cycle stages of, G1S, G2 division. And so she's got networks for each time point in dark and in light. And what she has found that at the transition of light and dark, based on her network analysis, she found that there are certain subnetworks in each temporal network. So these are temporal networks, but these are also spatiotemporal networks, because C2 has come up with a mechanism in her PhD to be able to spatially tag every node, here it's a gene, every gene to one location on the XY coordinates. And so these are hyper networks of spatiotemporal scale. And she found subnetworks here. If you look at the pre-dawn network, you will find that there is a group of genes that is active only at pre-dawn and nowhere else in the entire circadian cycle. She started looking at these genes and was able to identify novel regulators of CCM at pre-dawn, which are able to activate genes the moment light turns on out in the world. Okay, so I told you we are also very interested in artificial light at night stress, so ALAN stress. This kind of work might be useful for us in future. If any of my future students might ever be interested in picking up, carrying on C2's work, we'd like to, I know where next we would go to. So therefore, reformulating your problems, finding new things is part of what we do as part of as network analysis, okay? So yeah, we're now looking at network layouts and types, and how would you represent a network? So sometimes there are very common topologies in a network, and these common topologies can just be recognized when you look at it. The first one is a ring because it looks like a ring. The third one is a star because, duh, it looks like a star. And the last one is a fully connected network. Now, how would you think about, showing these to you on screen is one thing, but can you connect any of these topologies to a real network? For example, okay, let me tell you, a star network is when I am a stranger walking into your classroom today and I'm talking to, 20 of you, all of you are listening to me, but I don't, but each one of you now knows me and I know you, but you may not know each other because all of you are also coming in online from various places on the planet. And so therefore we form a social network of the topology star where all of us are connected to one hub, which may be your hosts, the Cphms, or it could be me because I'm speaking to you right here. So we form a star network. Alternatively, suppose I was your teacher and suppose you were 20 students in a classroom. So you're all sitting in your class, you're waiting for the teacher to walk in, you're thinking, how should I bunk? Should I hide under the table? Is she going to be absent? Is she going to ask me to take a test? But all of you are chatting with each other and discussing the arrival of a teacher. So what do all of you form? Because of the simple fact that all of you are sitting in a room and chatting with each other, it means that all of you know each other. And if you all know each other, that means all of you are connected to each other. So which of these topologies will you be representing if you're 20 students in a class who all know each other? Who's going to tell me in the chat box? And I am looking at the chat box. So it will be one of these networks that you will be, yes, Lavneesh, thank you. So somebody is listening and not thinking of bunking. So that means you got it right. Yes, that would be a fully connected network. Now, who's going to tell me, and on the chat box, not just now, when would you have a tree kind of network? When would you have a tree topology? And when would you have a bus topology? Think about it, put it in the chat box while I move on, okay? So there are different kinds of networks. And of course, the kinds of networks or the classification of networks depends upon the terms in which you're addressing those types. So if you're looking at relationships, then yes, networks could be classified by, what kind of relationship you're looking at. But a very general network typification is whether your network is a real network or not. Real world networks and random networks can be easily distinguished. And this is a first test that you should generally apply to your network whenever you've collated some kind of data and you're trying to see, should I do this analysis? Let's go ahead with network analysis. But before you begin, do the random network test and the small world network test. If your network, the degree distribution that we will talk about very soon, follows the power law, then you have a real network. But if it follows the Poisson degree distribution, as you can see on the right side of the screen over here, then it's very likely to be a random network. So don't trust a lot there. Now, the thing is, the difference between small world real networks and random networks, very layman way, is that the majority of nodes in a real network are having very few connections to other nodes, but some people are important. Some people are connected to everybody. So some nodes are hubs because they are connected to a lot of others, but most folks are poorer. So the rich keep getting richer and the poor get poorer, and that's the power law, right? So if the degree distribution follows the power law, then you've got a real network or a small world network up. In contrast, a random network is more homogeneous, where all the nodes have quite similar degrees. And this kind of a system where everybody's got equal power, more communist, I personally think that that is not a very stable system, because it's not robust to arbitrary node failure. Attacking a random network could potentially disrupt the connectivity within the network. There are arguments to that, but let's first try to just get to understand that the degree distribution is the main thing that separates them. And if you've got a real network, then you would have fewer hubs and more singletons on your network. Okay, and if you decide today after listening to me that you are kind of a little bit potentially interested in network visualization or checking whether you can use your data for network analysis, you have a countless barrage of free and licensed stuff out there, which would be, which would be available for you to use. And one of them that we use more of, I mean, we are using a lot of these. I'm personally a diehard R fan, and I use R all the time, and I think you should too. But Cytoscape is something that is much easier to pick. So R has a steeper learning curve, but Cytoscape is like you pick it today and you hit the ground rolling, literally. There are many, many other kinds of network visualization tools also, which are available to you at your disposal if you're interested in. I'll give you a quick primer now that I usually give to our PhD students, both here in India and in Cambridge. And that is when you begin with a very tiny small network. It's got about 10, 12 nodes, and all of them are connected by edges. So here is a network, a simple network. I want all of you to look at it carefully because I'm going to be asking questions. There are 10, 12 nodes. They're all named A, B, C, D, and so on. And there are these black colored lines that connect them, or edges. Now these edges could be, these nodes could be drugs connecting to receptors. These nodes could be proteins which are having a PPI. These nodes could be regulators with targets. These nodes could be people like you and me, with a friend's network, just about anything. We are going to try and talk about how this network and whether this network is robust against random node failure. And that brings in the whole idea of what I taught you about scale-free networks and random networks. Now, randomly, if you got rid of any single, chose any one single node to knock out of this system. So let's say this is the let's say this is the COVID virus spike protein network. And these are various proteins on the spike. So these are various, there's a spike region on the SARS-CoV-2, and it's got many different genes there. So let's say all of these people are on the spike region, and they're forming the network that we believe is very severely harming the host, okay? So your job as a researcher is to disrupt this network, this pathogen network. Which node would you choose to destroy as the researcher who's trying to disrupt this network? Would you choose A, for example, or would you choose something else? Alternatively, let's say this is a network of what is a good example? Yes, airport terminals, international airport terminals at IGI, or because next week I'm going to Finland, so Finnish Airways, or let's say this is the Helsinki airport, right? And in the Helsinki airport, you have these N numbers of terminals, and you are the security head of the Helsinki airport. It's an international airport. People come and people go, planes come and planes go, of course. And you need to protect it against a terrorist attack. So this time you have become the protector of the network. And if you were the protector of the network, which node would you put in all of your energies and all of your money and all of your staff and all of your guards? Which one would you guard with your life? So I see on the chat box a constant barrage of Ds, Ds, D, D, Ds. think again, sweethearts. Yes, absolutely correct in one way, that if you were wanting to disrupt the COVID network, or if this was the host pathogen network, you would think about disrupting D. But because you're also the pathogen, I put you in the shoes of the pathogen also. I made you the security guard of the airport network systems. So there, what would you be as a security guard? You're putting all of your security and money and your bets on D, because you know that the terrorist attack is going to happen on D. So as a researcher, I know the virus also knows that as a researcher, you're going to try and knock out D. So what does the virus do? It's smarter than you. Of course, this is a planet of microbes. Did you forget? It's not a planet of human beings. We live here at the mercy of microbes, of germs. And so the viruses, of course, not as smart as other germs, are smarter than you. Hello, welcome to the world. Yes, of course. So D is not the answer, because the virus knows that, smart that you are, you would come for D. And so it's protected D. It's the security officer who's protected D. Maninder, bless you. Gaurav, first time a student has said terminal nodes. Maninder, very nice. Sri Jenny also says D. Saumya says P. I want you to write in the chat box. Don't give me random numbers and think that somebody kahin na kahin to sach wala sahi wala nikal aayega. Of course, I'll have to give you marks. Sorry, won't work with me. You'll have to give me a reason for why Maninder thinks it's J, or why Saumya thinks it's P, or why Srabani thinks it's A, or why Gaurav thinks it's a peripheral node. Okay, now let me go on and explain to you a little bit more about robustness against random node failure. So what happens is, this is what Gaurav says. Gaurav says, kuch bhi peripheral hata lo. Ab peripheral node A knockout kar diya humne. What happened to the network? What happened to the network was, zilch, A died off somewhere and dropped off the circuit, right? What happened to B and C and D? Nothing. It is still intact. The network is still intact. This is a random node failure, and the system, being non-random and being small world, is robust to random node failure, such as that of peripheral nodes, which are not connected to other people and are just dropping off the face of the earth like little ball bearings. So there's another one for you. And there's another one for you. All of these peripheral nodes, ek ke baad ek, we are constantly removing, but because they are peripheral, because they are singletons, because they are not important to the system, they are not harming the system. And how do you know they are not harming the system? Because in network speak, or in network, how should I put this? In network fundamentals, as long as the system stays connected, as long as I'm a one-piece body, okay, you don't cut me into half or break me into disconnected fragments, I'm still able to connect with the rest of myself, and I'm able to communicate with the rest. So the communication flow is still going on. And so peripheral nodes, Gaurav, are not the answer. Let me look at Maninder. Good, very nice. Maninder says that it would be J. Now, targeted node failure would be D, which all of you said at the outset. And my answer to you is, targeted node failure is a theoretically correct answer, but not realistically, because again, it would have the host or the pathogen or the airport would have so many more protective aspects there. You would have a backup gene, you would have n number of mechanisms put in to protect D. And so your efforts as a terrorist, well, I should not call you that, your efforts as a researcher that is trying to disrupt the pathogen network here should be minimal, okay? Just like nature, kam se kam effort mein zyada se zyada value for money nikalo yaar. So then D is not the answer. And so Maninder, you get full points. J is the answer, because J is the bridge in this network, okay? And the moment you disrupt J, you're basically breaking the network into two pieces, which can no longer connect with each other or no longer communicate with each other. So what you should do as a researcher when you're doing network analysis, whether you're looking at a drug repositioning network, or whether you're looking at a ligand receptor atomic hydrogen bond network, or whether you're looking at the COVID pathogen network, you have to think about the nodes that are the bridging nodes. Now, what kind of centrality reflects the bridging nature of a node, and whoever's got the highest score on bridging is the one that you need to focus on. So full points to you, Maninder. I'm very happy somebody's got it, but I hope more of you can understand the concept of that. And that the fact that I was repeatedly telling you about communication flow, intuitively, what is communication flow? What do you mean when you say I am able to communicate to the rest of my being. Okay, so from the tip of my head to the tip of my toe, there is a direct connection of networks that can allow me to think that some ant just came and bit me on my toe. So essentially, what I'm talking to you about is the concept of shortest paths. So you could go from here to here, all the way from the other side of my head, but you could also go directly from here to here by connecting my ear to my nose. I mean, there's a shortest path to things, and there's also a ghoom ke chakkar, countless chakkars to think, right? So which one should you consider? Now, here's another network, and here are more questions to you. And so I'm looking at the chat box. The question is, you need to reach from node A in this network to node B. Okay, so how many steps would it take for you to reach from A to B? Let me answer that for you. If you have to reach from A to B, your first step will be to K. Then the second step will be here to S. It has been colored for you. This path, what I'm speaking out, the route from A to B is called path on a network. So the path from A to B could be A to K to S to R to X to B. And that is step 1, 2, 3, 4, and 5. And I hope you can see my, cursor? I think you can see my cursor. It's a bit glitchy, but it should be visible. Okay, so we have 5 steps. Can someone think of another way of reaching from A to B in more steps than 5, maybe 6 steps? And if yes, I want you to type it out in the chat box. How would you go from A to B, but not in 5 short steps, maybe in 6 steps? And there Shrabani has the answer already. Shrabani would take a detour at H. She would take a little tea shop and tea stall and stop there and have some chai, and then maybe move on. Is there something longer than 6, Shrabani? Can somebody tell me that you could reach from A to B in maybe 7 steps? Is that possible? Without repeating any nodes. So please don't tell me that I would go from A to X and then come all the way back to home to pick up my cell phone, which I forgot, and then I would go all the way back to school to B. That's not an option. Like the Leonard Euler question in the beginning of time in 1736, you too cannot touch the node twice. And so Madhusmita says, I have a 7-step route. Ravanish has already told that route. Absolutely correct. So you guys are now, what you're doing right now is telling me paths or shortest. I wouldn't call them shortest because you're finding longer and longer paths between nodes. And so therefore, all roads lead to Rome is a true fact. And so between any 2 nodes of a network, there are so many ways of reaching from one node to the other. And these different ways of reaching from A to B are called the paths from A to B. Okay. And so the way to be able to, can you think of a shorter path than 5? People have just told me paths of 6 length, paths of 7 length. Also paths of 8 and 9 you could think of, but can anyone think of paths smaller than 5, which I have colored here? I have colored in brown or red the path from A to B, which I thought was the shortest path. So if the network had to transfer a message or communicate from one end of the network to the other, what route would it take from A to B? It could have the option of taking so many different routes that all of you told me just now, but the network would always, always, always take the naturally shortest path, okay? So there is nothing shorter than 5, and so you can only reach from A to B in 5, and so therefore the shortest path between A to B is 5. But shortest path of 5 is just the path length. We call it the path length between nodes A and B. Who's going to tell me what is the path length between nodes Q and G? Can anyone see the screen? Can you see it large enough? You have got to tell me the path length between, okay, forget it, A to Z. Tell me the path length between A to Z. Is that also 5? Or is it something else? You've got to go from A all the way to Z, and Z is sitting here, okay? So who's going to tell me the path? Very interesting. Shabani says, can we break the bridge of SR and join AB? No, you can't touch the network because that's the data you've been given. You can't touch the data. That's the raw data. Okay, so within the raw data, you've got to now look for the paths. And yes, of course, if you broke this connection, you've completely disrupted your network. So in this network, if you were the if you were the researcher working on the pathogen network, your whole idea would have been to disrupt the interaction between S and R. Is it a protein-protein interaction? Is it a genetic interaction? Is it a regulatory interaction? Whatever it is, that's crucial for this network, because much of the communication in the network is happening in 3 modules. One module is this part, the other one is the B, the third one is down here. And to be able to disrupt this network fully, I would say you would have to break this edge and also, sweetheart, this edge. So 2 edges would totally disrupt this network. But for now, let's come back. The topic is Tikshana. Thank you for answering it. Anita has answered the root, but not the number. What is the path length? So when I say what is the path, shortest path, you've got to tell me, actually, you've got to tell me the path, which means Anita is right. And the path length is a separate thing. So the path length from A to Z is A, 1, 2, 3, 4, 5. It takes you 6 steps, Tikshana, not 7. So the longer paths would be 7 if you decided to go via X or even took a detour for a chai ki dukaan at T, then you would have longer paths. But the shortest path from A to C, in my view, is exactly 6 steps. The first is this guy, AKSR VYZ. AKSR VYZ, exactly what Anita has written. So the correct answer is 6 steps. And what I've now taught you is that all of these paths between any 2 nodes of the network, in all, how many shortest paths would there be? Between every pair of nodes, there is a number, which is the shortest path length of those, unique to those 2 pairs of nodes, I and J. And so therefore, how many potential shortest paths could you have in a network? N raised to the power of what? A large number. Okay, put it in the chat box if you can calculate it. But overall, the number of shortest paths in the network would be too many. And all of these shortest paths are called, there's a term, a scientific term, a network biology term for it, it's called geodesics. So of all the geodesics in the network, you calculated 2 for me, A to B and A to Z. Of all the geodesics in the network, which are actually the shortest paths between every 2 pairs of nodes in the network, the diameter of the network is what defines the speed of communication. And what's the diameter? Does anybody know the definition of diameter? Circle ka diameter, socho. Of all the paths or arcs in a circle, which one is the diameter? Sabse chota ki sabse lamba? So what is the diameter of all the chords, the longest? And so therefore, the longest of all geodesics, the longest of all shortest paths of a network is called the diameter of that network. So who's going to tell me the diameter of this network now? Let's see. For knowing the diameter of this network, you would have to really put your brain into finding every single pairs of nodes and then looking for what is the shortest path between those pairs of nodes and then lining up all the shortest paths and then finding out what is the longest or the shortest of all the paths. Itna sab karne ki zarurat nahi hai, because this is a small network and you can just with your eyes see which are the 2 nodes which are farthest apart. So Anita says that the longest path of this network or the diameter of this network is 6. Does everybody agree with that? Or can you find another shortest path between any 2 nodes, which is 7? Think, and think carefully. And I would still right now try to put my bets on Anita, because I think, okay, 5 diameter, no, 5 cannot be, it's 6. Soumya says 6, I'm sorry, Anita said 5. So the diameter cannot be 5, Anita, because there is another 2 pairs of nodes which are farther away from A and as are A and B. So A and B have the shortest path length of 5, but Z and V, if you look at Z and V, which are both part of the same network, have a distance of 6. Similarly, A and Z have a network distance of 6, and that's the shortest distance. And so therefore, always keep in mind that the longest of the shortest paths is your diameter. And the diameter is the maximum length that it would take for every node to be immediately communicating information to the other node. Okay, so the concept of diameter is extremely important. And there's a last question on this slide, which I think you can't see because... Okay, we need to mute somebody's Dell. Right, let's move on. There's not a lot of time. Uncle Dell, you need to mute yourself. Okay, so network diameter, there's an exercise. We've done the exercise together. Thank you. Network partites, I'm going to skip over this because I think I don't have a lot of time to teach you so much, but I do want you to know that as we talked about types of networks, there are also network directionality. Networks could be directed or undirected. Can someone give me an example of a directed network? I'll tell you. A regulator and target network is directed. A teacher-student network is directed. Now, similarly, what would be an undirected network in which the nodes have no direction? That means they are bidirectional, essentially. Well, yeah. So can you think of an undirected network in which the relationship works both ways? It's a two-way traffic. And comparatively, a directional network is a one-way traffic. Okay, so who's going to give me? Yes, Sonali, friendship is an undirected network, a friendship network. Facebook, all of your Facebook friends are the directed networks. Twitter, on the other hand, is a directed, not an undirected one, because you follow people and people don't follow you back and so on. Okay, so edges could be directed, could be undirected, and that would be the network formed by those edges. And then there could be weights on these edges. Some edges. So if you've got 5 friends, do you think you would be equally, you would be a communist, yes, and you would say, oh, I love my friends equally. But me, I am not that. I like some friends more than other friends. So how does that work? I have only some friends who I always tell that I'm giving a lecture, why don't you come and listen? I don't tell that to everybody. So there is a weighting to the edges associated, right? Some edges have more weight and other edges have slightly less weight. So who's going to give me an example of a weighted network or a weighted edge network where some edges have greater weight and others have lesser? Dikshana, you need to unmute yourself, sweetheart. I mean, you need to mute yourself, Dikshana. So in the meantime, who's going to tell me, how about a weighted network, a parent-child network? Is that a weighted network? Oh, no, no, no. I have 2 kids and my God, I don't don't even dare suggest a weightage between them because they will come back to bite me as nobody's business. So I would say a weighted network would be a co-expression network, a weighted co-expression network in which 2 genes potentially have more, are expressed with each other with a higher correlation coefficient than maybe another 2 genes and so on, okay? Protein-protein interaction networks. Now, protein-protein interaction could be a bi-directional interaction because physically both of them are touching each other, right? A ligand-drug interaction. Think about how these would be directional and whether they could be weighted. And you have to give me an example in the chat box about directional and weighted networks, okay? We are going to be running into, I need to ask the hosts because it looks like it's already 11. Did we have a time set to this talk so I could potentially wrap up quickly, or do you think I have a few minutes? Kadambari, you've got to tell me.
We can wrap up in a few minutes. That's fine.
Let's do that. Let's do that. Yeah. So I'm going to skip over graph matrices and centralities. I'm going to, in general, what I've already taught you about is a degree is that the degree is the centrality that lets you see how many different nodes you have. Scale freeness is something we've talked about. Transitivity is the number of modules that we saw together. Shortest paths you've already seen with me. Community senses would come from partitioning a network. You can find the modules in a network. When you're doing network analysis, you should first do a visualization. Second, you should do centralities. Third, you should do network partitions. And how do you do network partitions? You do based on, in general, connectivity. Okay, so your clusters, that means you're just going to make your whole furball of a network become meaningful by making clusters. And these clusters have to have the property or criteria satisfaction for homogeneity and being able to separate from within a cluster. All the genes or all the nodes have to be similar. Outside of a cluster, they can be different. Okay, so your aim right now is to find a highly connected subgraph. Let us do this experiment together and then let's be done with the day. Okay. So within the cluster, like my student C2 found out, you need to identify highly connected subgraphs. We call them HCS or HCG, and we find them based on edge connectivity. So you need to know what is edge connectivity. Edge connectivity is the minimum number of edges whose removal will result in a disconnected graph. So let's say you have a graph, you and I are connected, and there's a third person listening to us. So we make a triangle. How many edges would you have to remove from the 3 of us to disconnect the 3 of us? Okay, so for a graph, if the edge connectivity is 1, then it's called a 1 connected graph. You're going to tell me now, this is an example of a graph which is ABCD 4 nodes. What is its edge connectivity? Okay, how many nodes, how many edges will you have to remove to break this graph into two pieces? Removing one edge is not going to break this graph into two pieces. And what about this one? Who's going to tell me the edge? Sarabani says the edge connectivity of the graph 1 is 2. And what's the edge connectivity of the graph 2, Sarabani? Can anybody tell me how many edges you'll have to remove on the right side graph to be able to get a disconnected graph? The answer is that the first one, Sarabani, is right. The graph disconnectivity or the connectivity is 2. And for the second one, it is 3. Rita, no, not 4, my dear, just 3. Sonali is right. If you just remove 3 edges, you've disconnected this one. You remove anything around B and B is fallen off the grid, okay? So similarly, a cut or a min cut is a cut denoted by S, which is a set of edges whose removal disconnects the graph. So what you basically found is the cut, okay? Now here is an example of the same network, and how would you do the cut? The cut of this B will disconnect B, the cut at C will disconnect C, and so on, okay? So these are called cuts, and the min cut and the disconnectivity are both fundamentals that will allow you to identify a subgraph. Now, what is the rule for the subgraph? For a graph whose vertices are more than 1, you will call it a highly connected graph only if its edge connectivity is greater than n by 2. N means the total number of nodes, and you divide it by half. If your connectivity is greater than that, you've got a highly connected graph. Otherwise, no. So remember the rule. For a graph with more than 1 nodes, if it's a highly connected graph, if and only if its edge connectivity is greater than n by 2. Now, here is your graph. What is the connectivity? What is the edge connectivity? Who's going to tell me how many edges you have to remove to get a disconnected graph? Nirupama, very good. I'm asking a question on this graph right now. You're going to tell me whether the connectivity, now I will tell you the answer. You just need to remove this one edge, okay? The moment you remove this one edge, you're going to have a disconnected graph, and therefore the edge connectivity is 1. And the number of nodes is how many? 5. So is this a highly connected graph based on the rule that I taught you? Mahadev, 5 is not the answer to connectivity. Connectivity is 1. The number of nodes is 5. Rita and Nirupama and Sarabani, they believe that this is not a highly connected graph. All 3 of them are right. Absolutely brilliant. This is not a highly connected graph because the edge connectivity is not greater than n by 2. Now, here is your second example. Are you going to tell me whether this is a highly connected graph or not? Yes or no is all I want on the chat box. Tikshana says no, but Varsha says yes, and Sonali says yes, absolutely brilliant. The edge connectivity of this graph is 3. The total number of nodes is 4, and this is a highly connected graph because 3 is bigger than 4 by 2. Okay, now this is how your big furball looks, and your job is to partition this network. How would you go about partitioning the network? Find the edge connectivity, which are the edges that you're going to remove to disconnect this graph. There you go. First of all, this is 2. So if it is 2, is this a highly connected graph? The answer is no, it is not, because 2 is not bigger than n by 2. And so therefore, because your answer is no, you will actually cut those 2 edges and remove them. The moment you remove them, what are you left with? You're left with the left side and you're left with the right side. And continue there. Is the left side a highly connected graph? Is the right side a highly connected graph? The left side is, yes, but the right side is not. And therefore, you will go ahead and cut it further. And the moment you cut it further, you find 3 highly connected graphs, all following the rules of the connectivity being greater than n by 2. And so finally, your clustering algorithm has now found 3 clusters on this network, which started as a furball, and you can keep going on and until you find only highly connected subgraphs. And the moment you find them, you've done your partitioning, your network partitioning is done. Lesson over. There are many other clustering mechanisms, which I'm not going to teach you today, but usually I do teach MCL and flow-based clustering and so on. But I'm going to stop all of that and maybe talk to you another time. For now, I just want to say, do think about networks. It's very, very good to know networks, okay? And it's very interesting to work with networks. So thank you very much for listening to me. and you should pick up a lot of other data skills in R, or join my protocols from home. I keep tweeting about them all the time, and I'm at Gillian B. And of course, later today, we are working, we're showing you what young scientists can do in the age of knowledge neocolonialism. That too is happening at the Climate Hackathon. So big thanks for listening to me and answering all my questions. And I'm willing to take more questions if you have any.
Thank you, Dr. Yadav, for this wonderful, wonderful webinar. I think I can speak on everyone's behalf that this has been one of the most engaging webinars I personally have attended. I think everyone had a great time. So thank you for being with us today. If anyone has any questions, please drop them in the chat box. Maybe we'll give everyone a few minutes to send in their questions, and then we can have a short Q&A session with Dr. Yadav. And if you would like to, if you would like to talk about anything else that you're currently doing, ma'am, you can go ahead while we wait for the questions.
Yes, sure, absolutely. I see a lot of good webinar, happy webinar, interesting webinar. Arey, abhi to maine khatam hi nahi kiya tha. Next time I will finish the whole talk and then you tell me how interesting it is. But for now, I see an interesting question by Sonali, where the visibility, is success or visibility more dependent on networks rather than the work itself? Mona Lisa as opposed to other paintings by Da Vinci. That answers the question automatically, Sonali, meaning you know the answer. And what you know and what you've seen with Da Vinci is also exactly, absolutely more true in the world as we live in. Much depends upon visibility. You could do billions of good science and good work if it's not visible, if it's not you. So citizens do not want your papers. Citizens don't want to know what, what thought went into how much of a genius you are. They want to see how your work is connecting to society. And I've been saying that quite a lot. Citizens want science, citizens want knowledge. They don't want your papers. But we as scientists always think, my work is just to get my APAR signed and my score there and my papers in. I don't think that's the right way of going ahead. And I think it's very, very, very important for you to step out of your little 4-office wall and go out and meet people and tell them what you're doing and let them know what you're doing. Because if they don't know what you're doing, then tomorrow that's going to come back to bite you when the government asks you or your funding agency asks you, "Bhai, tumne kya kara?" You can't any longer submit your list of thick sheaf of papers, because that is garbage as long as nobody understands it. And so therefore I think visibility is far, far, far more important. I see that a lot of people understood the concept, and I'm very happy to know that you did. And the whole idea of teaching is that you should understand the concepts, not how to. So these are non-trivial things. Clustering a network, partitioning a network is non-trivial. And yet the fact that you can understand it and you have actually done partitioning yourself on a network should tell you that you can do it too. Okay, so pick it up, pick up the skills and do it yourself. Start working on it today itself. Pick up a Cytoscape tutorial. Everything is free on the web. All right. So, where do you need, you can pick up more on network skills by looking at the freely available Cytoscape tutorials on the world. But if you prefer R, then there's countless more tutorials on R out there in the public domain. Google Bhagwan se puchiye, everything is out there. I would say in general, I mean, Cphms is recording my talk and I appreciate that. It will be on YouTube sometime, but I personally don't think watching somebody's lecture on YouTube later is the way you're going to learn something. You have to be in the class with me. You have to answer the questions when I ask them, because then I will know that you've not answered it correct and I can tell you where you were wrong. But if you listen to the lecture later, I don't know whether you're listening. You don't know whether you're listening. Like my son, you might go to sleep in the class and then during that lecture. And, we don't want a lullaby. We want you to be listening to what to do. The softwares, Rita, I've already given you a whole list of softwares on that page, which I told you, but I can tell you offhand are iGraph, SNE, SNA, packages, also Cytoscape. Just begin with Cytoscape and you've got at least 1,000 plugins within Cytoscape to look at, biomedical data, drug ligand data, plant data, regulatory network data, miRNA data. Godforsaken, every data under the planet is in a package or a plugin of Cytoscape. So Cytoscape was built at the University of San Diego, and it's a big consortium of academics who built it together. And so therefore, please please go and use Cytoscape. I also see a question saying protocols from home. Vipra, join me on Twitter. So whenever I give a talk, I like to tweet about it. If I'm giving it on a Sunday or a Saturday, now next week I'm traveling, but I know that after I travel and after I reach London, I'm going to be free for 10 days in the Dussehra holiday. So I will be doing a lot of lectures at that time, basically are. So if you follow me on Twitter, you might be able to see some more of this. What else? What else? Any more questions? Thank you. Welcome. Welcome. Yes. No. Nobody else. If you have a class of mother-in-law, daughter-in-law, and the daughter, Sarabani, very interesting. Kaun sa network tha wo? Tree, or was it a fully connected network? Okay. No ragging here. What else? Good answers, a lot of answers. Topology might be related to computers. Lavnish, good. I was wondering who would give me the answer on topology with computers. So I'm also the systems administrator at NIPGR. And we started this LAN over here, the IT LAN, based on a bus topology. And then we, over the years of revamping and fixing the institute again and again, we went into from bus to mesh topology, from there to tree topology. And finally, right now, we are in a chaos topology. We don't know what the network is because it's been revamped hundreds of times. So yes, it all began, the topology business began with computers. You're absolutely right. Walled office consisting of computers, host of a star topology connected to one student, computer hubs, tree showing the relationships. Dendrograms are tree topologies. Anybody else? I think everything's good. I think we've got a good series of messages. Okay, that's about it. Thank you.
Hey. Thank you again, Dr. Yadav. I think, so for all the participants, we'll be sending out an email after this once we upload the webinar to YouTube. And hopefully we'll get maybe some more information on how you can join Dr. Yadav's classes, all of her social media details. You'll be getting an email. So just keep looking at your inboxes. We'll send you that soon. Thank you again for this wonderful session. Thanks for everyone for joining us this Saturday morning. Stay tuned. We'll have a lot of more interesting webinars coming soon. Thank you again for joining and have a great day, everyone.
Thank you, ma'am.
Bye. Goodbye.
Thank you.
Bye-bye.
Bye.